All right, so back to our session. We're going to have now Jennifer Panagoulias. I hope I pronounce your name correctly. Jennifer is a regulatory affair expert. She's the COO of AS2 Bio, so we're going to hear about that, I hope, and the head of regulatory and policy of FAST US. She's co-director of ABOM. She's an expert for many years, has been working in drug development, particularly for the treatment of rare diseases, as well as working with regulatory agencies worldwide. Jennifer is going to give us an update on gene therapy programs, particularly on AAV, adeno-associated virus, and ex-vivo gene therapy. Jennifer? Oh, here she is. Thank you.
Thank you so much. Thank you, Benedetta, and to FAST Italy for inviting me to speak to you today. And I'm going to talk about some of the exciting programs at the accelerator that Allison mentioned. These are my disclosures. And one important thing that isn't listed here that I want to share, and that is that I'm an aunt of a 23-year-old young woman who lives with Angelman syndrome, and that is what brought me to FAST. So I'm going to talk a little bit about the accelerator, and I'll start with FAST. And Allison showed you the roadmap that FAST created for development of therapeutics. And what's really important here is that in deciding to start an accelerator, it really ties in with their vision to go from science to the patient, or bench to bedside. And in developing the roadmap, Allison had the vision that in funding research, and FAST US has invested $50 million in research programs since its inception in 2008, that you had to not just have the money to fund scientists to create discoveries, but also to give them resources to take those discoveries from the bench through development and into clinical trials. And so that means animal models and cell lines and endpoints, meaning how we measure change in patients. And then ultimately, that gave rise to the development of the accelerator, which is AS2 Bio. And so what we're going to focus on today is really what those accelerator companies are doing right now with research programs for Angelman syndrome. And this is who we are. This is AS2 Bio and our team that we've put together that has deep experience in development of rare disease therapies, and in particular gene therapies. And so this is the company that manages the development programs that I'm going to talk to you about today. And we've hired a great team, and we have a few more people that we plan to hire this year to help us in this mission. So the programs that AS2 is managing are shown here, starting with MVX-220, and I'm going to talk about that. It's an AAV gene therapy for Angelman syndrome, and it's being run under a company called Mavericks Bio. And in the middle, we have UB-cell, which is another kind of gene therapy that's in a stem cell, a blood stem cell, and that's under a company called Transformatics. And then we have a third program, which is a CRISPR, gene editing, and Allison explained that in her talk. And that's being investigated by a company called Courageous, and that's under a grant from the US NIH with Yale University and Rush for $40 million to develop that program. So that's a great investment by the US government in the development of a CRISPR. But what we're going to focus on are really these two gene therapy programs. So I'm going to start at the top, which is what is gene therapy? And it's essentially using a virus to deliver a gene of interest. And in this case for Angelman, it's using the virus, because they do a good job of getting into cells, to carry the payload, which is in this case UBE3A. So it's a means to provide a healthy copy of a gene into the body. And there's different ways that gene therapy can be delivered. One of the ways shown on the left is called ex-vivo. And in this case, what you do is you take a cell from the patient's body, like a blood cell, take it out, put the vector inside of it, and then you transplant that cell back into the patient. And there it delivers the UBE3A payload. The other type is when you actually just directly put the vector into the body through an injection, and that's called in vivo. So we're going to talk about that right now for a minute. So essentially what you're doing is taking the gene, here UBE3A, and you're putting it in a viral vector, and then you inject that directly into the body. You could do it by IV administration, or you could directly inject it right into the brain or into the cerebrospinal fluid, the CSF. And then once that vector is inside the body, the virus gets into cells and has the instructions to make the missing protein, in this case UBE3A. And what we're going to talk about today is an AAV virus, and these have been used in other research programs and even in products that are approved like Zolgensma for spinal muscular atrophy. So it's going to deliver a functional copy of the UBE3A gene. So the program that Mavericks has responsibility for is called MVX-220, and it's a UBE3A AAV gene replacement therapy. And that's shown on the left, the human UBE3A gene. And it's actually been studied already in mice and in monkeys, non-human primates, and I'm going to show you some of those data. And the intent of those studies was really to prepare for clinical trials. So this has a lot of information, but let's start in the middle. So one of the things that has been investigated with MVX-220 is its distribution into the CNS. And the plan for this program is to inject it into the cisterna magna, and it's called ICM administration, and it's right here. At the base of the skull is a small spot where there's a reservoir of that CSF fluid. And because gene therapy is really only intended to be administered a single time, so not repeatedly like an ASO, this procedure can be performed even though it's a little tricky because you only have to do it once. And so that's shown on the top, that the base of the skull with the injection into the ICM. You may have also heard about lumbar puncture, which is another way to get drug into the CSF, and that's being done in the ASO studies. And in the box on the left, if you look at the red and blue, when the team at University of Pennsylvania investigated the delivery of gene therapy, they looked to see, was it better if we administered it by ICM or by lumbar puncture? And you can see you get a lot more in red into the brain if you put it into the cisterna magna. And so the plan is for this program to pursue the ICM route. So several studies were performed in the Angelman mouse, and I'm going to take you through some of the results. And these studies were intended to support the plan for clinical trials. And in particular, this study was really intended to look at different dose levels to understand what's the best dose level to use in human trials to get a response. And so they used newborn Angelman syndrome mice. They looked at a low dose, a mid-dose one, a little bit higher, mid-dose two, and then a high dose. And basically they administered it to animals at birth, and then they followed them. They looked at their weight, they looked at their clinical scores, which is really a means to look at safety in the mouse, and then they looked at behaviors in this Angelman syndrome mouse. And the Angelman syndrome mouse has some characteristic behaviors that are thought to tell us a little bit about how the drug would work in a human. So here we have three things that were evaluated in the Angelman syndrome mouse. One is rotarod, and that's basically how the mouse's motor function is and how they perform running on a twirling rod, and do they fall off faster or slower. They looked at open field, which is really an analysis of their motor function and anxiety. And then they looked at nest building, which is an innate behavior to the Angelman mouse that's really thought to reflect both their motor skills and cognition. And what you can see here is that in black are wild type or healthy mice. In red are untreated Angelman syndrome mouse. And then kind of in the middle are all those different colors. And those are the dose levels of MVX-220 that were administered to the mouse. And so what you can see is that, generally speaking, MVX-220 treatment improved their performance on the rotarod, in the open field test, and in nest building. And if you look at the box on the far right, essentially the higher doses of vector did a better job in improving that nest building capacity in the Angelman mouse. And in some cases, it actually overlapped with the performance of the healthy mouse, the wild type. They also looked at safety parameters. They looked at body weight, but body weight really is a reflection of Angelman syndrome because, as you know, some children with Angelman syndrome have hyperphagia or food-related behaviors. And the Angelman mouse, shown in red, was a little bit heavier than wild-type mice. So they saw that the vector improved the weight phenotype in mice. And then on the bottom, they looked at brain weights and the clinical score, which are really reflective of safety. And the vector really didn't do much to change brain weights in the mouse, but largely the clinical scores were all the same between treated and untreated mice, meaning that there was no safety concern there. And they also looked at labs, and they looked at histopathology, meaning they looked at the tissue after treatment to make sure there was no strange findings there. And essentially, it was a very positive safety review and suggested that this could move forward into clinical trials. So this vector was also tested in monkeys, where they looked at distribution and they looked at the safety. And this is a real rigorous evaluation, generally in a toxicology study of a monkey. And they looked at 2 doses, a high and a low dose. And what you could see here on the left in the pink bars is that the vector was distributed throughout tissues of the brain. And in the second panel where it says HUBE3A1, you could see that the vector was producing UBE3A in tissue, importantly in tissues in the brain. On the right-hand side, you can see at the high dose, these are pictures of brain cells where you can see in red that UBE3A is being expressed in tissue. A safety analysis was performed here as well, where they looked at laboratory findings, clinical findings, and again, histopathology, so they looked at the tissue throughout the body. And these data were supportive of taking the vector forward into clinical trials. So really, in summary, MVX-220 vector showed improvements in some of the functions in the Angelman syndrome mouse. It was expressed in the brain. When you put it into a larger animal, a monkey, you could see that it expressed UBE3A in tissues that you want it to be in the brain. And it was safe overall and generally well tolerated. So now we're going to talk about the second program, which is another kind of gene therapy, but a little bit different. It's ex-vivo. So again, it's administered outside of the body. And by this, I mean you still take a virus and you insert the UBE3A gene into it. But what you do is you take a stem cell, a blood stem cell from a patient, and this can be taken by IV. It gets filtered out. And then you put the vector into that cell, and you transplant those stem cells back into the body. And essentially, they differentiate into blood cells. Those blood cells go into all throughout the body. Some go across the blood-brain barrier and become immune cells in the brain. And those immune cells express UBE3A that can be taken up by neurons of the brain. So it's just a different way to administer a functional copy of the UBE3A gene. So in this program, what we did was we took adult mice. So first we took blood cells from the mice, put the vector inside of it, and then injected it or transplanted it into Angelman syndrome adult mice. And so this is important because this study used adults as opposed to the other study I showed you, which used newborn mice. To give a transplant, whether it's a human or in a mouse, you have to deplete or remove some of the existing blood cells because you have to really make space to put new cells in. And they use an agent called busulfan, which is a chemotherapy, to reduce the number of blood cells. And so you have to make some space so that the new cells come in and can grow and proliferate throughout the body. So these mice were followed for a similar battery of behavioral tests, and they also looked at safety in these mice. And what's important here is this gene therapy is designed to make UBE3A, but because it's in a blood cell, it also has a signal, it's called a secretion signal, to make it leave that blood cell so that it can be taken up by neurons. And this is a busy slide. It has a lot of information. But what's really important is what's shown in the box. And that is we use two different kinds of vectors to treat mice, both that contain the UBE3A, and we compared them to healthy animals, wild type, Angelman syndrome mice that didn't get a treatment, and Angelman syndrome mice that got a mock transplant, so a sham. They got a transplant, but it didn't have a vector with UBE3A inside of it. So we looked at all the different conditions. And what's really important here to follow on the slides that I'm going to show you is that the blue and the red are Angelman mice treated with the gene therapy. And what you can see here, this is the open field test. Again, looking at anxiety and motor performance. And largely what you can see is the blue and the red do better than the brown and the gray, and those are untreated mice or mock-treated mice. And when you look at their results compared to healthy mice, they look very similar. So their performance is very much improved in the treated Angelman mice, and it looks good almost like healthy mice. When we looked at novel object recognition, this is a different test that looks at cognition, where, again, the blue and the red here are showing how much time the mouse spends with the new object as compared to the old object. And it's really meant to understand if they can discriminate between what's new and what's old. And if you look on the far right, the Angelman mice that weren't treated can't really tell the difference. They treat both objects the same. So again, the treated mice look good, and they look somewhat similar to the wild-type mice. This is a balance beam, which is a measure of function. And this is from the video that Allison showed you, where you had the mouse run across quickly. That was the wild type. And the treated mice look very similar, but the Angelman untreated fell off the beam. And what you can see here on the far right is at the skinniest beam, the 10.5 millimeter beam, the smallest beam that's hardest to cross, again, the treated mice did better than the untreated mice and looked very similar to wild type mice. And this is a really important slide because it's looking at UBE3A expression in the mice. So if you look at the top row, which says AS2 HET2, this is the Angelman mouse that was not treated. And if you look at the third row that says UBE3A, you can see there's pretty much no green in that panel, and that's because the Angelman syndrome mouse isn't really making any UBE3A. But in the three boxes below, the wild type, and EFS, HPGK, you have untreated, and then the bottom two are mice treated with the viral vector. You can see that they're making UB3A, and it looks very similar. And most importantly, on the far right, where it says merge, you're looking at a combination of neurons, their expression of UB3A, and the immune cells. So essentially what this shows you is that the immune cells, so the blood cells that we transplant in, are differentiating into immune cells. Those immune cells are going into the brain of these mice. They're secreting UBE3A, and it's getting to the neurons. And so this is really important because it's showing mechanistically that you can basically deliver the gene therapy into the blood cell, and it will get to where you want it to go. And that's really evidenced in the results that we saw where the mice had improved behaviors. And so what's really exciting about this program is that last week, or at the very end of January, the team that had worked on the development of these vectors at UCLA received a grant from an institute in California for $5.8 million that will really fund further development of this gene therapy. So this is really great news because in the current financial environment, sometimes it's hard to get investment in programs, but this will essentially pay for toxicology studies and manufacturing development to take this forward into a clinical trial. And so to sum up, our next steps at AS2 Bio, Mavericks and Transformatics, is to take these forward. Most importantly, we expect to announce the initiation of a phase 1/2 first-in-human clinical trial of MVX-220 later this year. And when we do, we will have a webinar with FAST to provide details on the specifics of that study. And we plan to initiate IND-enabling studies of UB-cell, the lentiviral gene therapy, to take that forward eventually into a clinical trial in humans. And I just thank you with acknowledgments to the teams at UPenn and UCLA that have worked so diligently on the development of these programs.
Thank you, Jennifer. Wonderful presentation. And the data are very exciting. And I'm sure they're going to raise a few questions because they are very new as a way to treat Angelman syndrome. I have a few, but first from the floor. Questions? Don't be shy. Over there.
Oh, I don't have the...
I'll translate.
Purtroppo l'inglese non è la mia seconda lingua. Allora, la domanda è questa. Mi sembra aver capito, sono un genitore, che per i topi adulti, insomma, le prospettive sono molto molto interessanti. Quindi per i pazienti adulti, io sono papà di 2 ragazzi, uno di 35 e l'altro di 24, e l'altra di 24. Che tempi, cioè i tempi canonici, diciamo, per poter poi arrivare al farmaco? La domanda è molto, molto, come dire, diretta, insomma, si capisce, no? L'importanza. Quali potranno essere?
Traduco, sì, sì. I'm going to translate, but you can also start wiring yourself. So he's a parent of 2 adults, Angelman, 24 and 35, I believe. And he was very interested in the data on adult mice. And he's asking, what is the timing from getting to these data to treatments for adult Angelman?
Well, I can say that there are adults that are being treated with the oligonucleotide therapies. I know the phase 1-2 study from Ionis included adult patients, and they're here, and I think they'll be talking to us about their plan for other trials. For the lentiviral gene therapy, we hope to get into clinical trials, but it will take some time. It takes sometimes a year or more to run the animal studies. But our hope would be eventually to include adults in those trials. From my own personal family experience, it's very important to me to understand the safety and potential effects of therapies in adults, because I don't want my own niece to be left behind.
Thank you, Jennifer, for your brilliant presentation. I'm just wondering if there are some data comparing the level of distribution of UBE3A in brain between the ASO approach to the AAV approach. So if there are some data that compare the level of distribution of UBE3A in brain cells.
I'm not aware of any direct comparisons, but I'll ask Allison if you know.
So nothing has been head-to-head in the same experiment, but there's mouse data from different labs, but highly reputable labs, looking at ASO in the same age versus AAV in the same age, so newborn versus adult. And basically, when they're very similar, ASO and AAV, if they're delivered into the CSF. The issue in humans that is expected is what is the distribution from a lumbar puncture, which you have to do for repeat dosing, versus ICM in being closer to the brain to bathe the brain. That's the big question. And that experiment, because they're different size molecules, has not been done head-to-head to understand that. When you look at the number of neurons exposed with ASO in mice to AAV with mice, it does look like the exposure of AAV in mice is higher than ASO in mice. But a mouse and a human are absolutely not the same. So it's very hard to say. Repeat dosing can compound effect in a beneficial way, where AAV, a single dose, you get what you get. And so it's to be determined. And eventually, hopefully, it will be done head-to-head in a larger animal, but it has not been done.
Thank you.
Other questions?
Thank you for your presentation. So my question is about transgene expression within the brain. We don't know the level, so is there any plan to control this expression, to prevent overexpression or expression leading to pathological stuff? See what I mean?
I do understand, yes. I think that this is a question that often comes up from the regulatory authorities, in particular, to make sure that we're not overexpressing. And so sometimes in toxicology studies, they ask us to evaluate neurological behaviors to ensure that there are no aberrant or unexpected behaviors that might relate to overexpression. So we haven't seen that, and I think that is likely because we're not really close to the true, levels of UBE3A that the body makes, even with gene therapies. We probably just don't express as much as you would traditionally.
Thank you.
More questions? While you're thinking, I have a couple of technical questions, actually. So one is to understand what's known about how these cells reach neurons. Because is it the virus entering neurons transported by immune cells in the brain? What exactly is known? Because the protein doesn't get into a cell unless there is a positive transporting mechanism.
Yeah.
So I'm a little, questioning the steps. Sorry to go a little bit in details, but I think this is important for thinking about the next step in research.
It is, I agree. That the lentiviral vector has been engineered to include a secretion signal, but also the protein has glycosylation sites. And so the intent was that the immune cell could secrete UBE3A, and then it had carbohydrate structures that would allow it to be taken up, including by M6P receptors.
So there is a modification that I'm particularly interested in that because I'm going to talk about cation-independent mannose-6-phosphate receptor later. So there is an engineered piece that will bind to receptors that should internalize.
Yeah.
And how much is the transport in the brain, given that the blood-brain barrier is really stopping the cells to get into the brain in normal conditions? I'm not sure in Angelman syndrome the blood-brain barriers has been studied a lot, so maybe is less effective and therefore leaves more room to enter into the brain. Is something known about that?
We looked at levels of engraftment of the vector in different tissues, and we saw that the engraftment in the brain was much lower than in other tissues. But despite that, all of the phenotypes were corrected in the mouse.
Oh, I agree. The behavior is really nice. Yeah, nice data.
So to your point, yes, the levels are lower, but it seems at those levels we were getting good behavioral changes. And I didn't show the EEG, but that also looked quite good.
And it goes throughout the brain?
Yeah. Yes.
In multiple neurons?
Yes, based on the staining, yes.
Okay.
I can also add a little bit
Yeah.
to that.
Yeah.
Just from the crossing of the blood-brain barrier. So, the beautiful examples of this are metachromatic leukodystrophy, adrenoleukodystrophy, cystinosis, MPS, and all of those. In Angelman, the blood-brain barrier is thought to be perfectly intact. There's no inflammation and reason to believe it's not intact. Similar, what you see in wild-type mice that are treated with these types of therapies, it beautifully crosses the blood-brain barrier as microglial cells. They become microglial cells. So essentially it's macrophage, it's monocytes becoming macrophages, becoming microglia that populate the brain. But generally microglia naturally are in the brain in much less numbers than macrophages might be in the skin or other places that they go outside of the systemic circulation. So the number of microglia that are there are considered appropriate for a neurotypical brain, seeing it in the Angelman mouse as well. The thing is that they're surrounding most neurons, and they secrete around all of the neurons. So I think the reason we're seeing so much more benefit with this program than others is because it crosses the deep brain structures of the blood-brain barrier compared to just those bathed in the CNS. And because it's ex vivo, there's no virus that's systemic. So it's just in the CD34 cells that go into all of the other progeny that become microglia once they cross naturally. But even in all of these other programs, MLD, CALD, cystinosis in humans, where they're degenerative, they're completely curative and not causing any disease in those individuals. Seeing engraftment at maybe 100 times less than what you would see systemically, it still bathes the neurons enough to actually fully rescue the phenotype in humans with these other degenerative disorders. So there's a real movement in the field, in the CNS field of genetic disorders, to, if you can get a cross-correction effect of an enzyme, that this may be one of the best ways to get to all the neurons. And that's a movement that's been happening over the last 5 years.
You know, I'm sure this is very exciting, but I think a number of steps are still not clear. Forgive me if I'm stressing that, because, you know, the immune system is not going naturally into the brain. That's not what in the normal condition happens in a developed brain. Maybe during development there is more of these movements. And also microglia is developed during early phase of development, and then you work with the microglia you have in the brain. So if there is an insult, microglia becomes activated, reactive microglia, but that's not the normal microglia. So there could be macrophages. Anyway, just to say, this is very technical, but just to say that there are a number of steps that are going to be understood,
Sure.
and that's very important.
Yes.
The data are really very exciting. Thank you, Jennifer.
Thanks.
Any other question, by the way, while we had this discussion? Yes.
Volevo chiedere, questo trattamento che più in là si farà, lo possono fare qualsiasi bambino Angelman, piccolo, adulto, ma con qualsiasi variante PD delezione, perché attualmente gli ASO li stanno applicando solo sui bambini che hanno la delezione. Quindi più là ci sarà qualcosa da aspettarsi anche per le altre varianti oppure no? Grazie.
Yes, that is a good question. And our expectation is that this could be used in all genotypes because it is not targeting the father's gene and trying to unsilence that gene. So there's less of a risk of overexpression in some of the genotypes like UPD.
Any more questions? One there and one there.
Hi, Jennifer.
Hi.
I'm wondering if we're looking also if some cells are being changed that we do not wish need to be changed, and that we're only targeting the neurons we want, and whether it's part of the study to see how many are impacted or not.
That's something that we would look at in terms of the toxicology findings, right? If there's any off-target effects, so that will have to be carefully studied.
Thank you, Jennifer. Question regarding IT versus ICM. Which one do you think it's going to be safer, learning from Zolgensma also from the past and from other gene therapies? What do you think about that?
With ICM, it was, sorry, I don't know if you can put that slide back up, or you don't have to, but the team at Penn saw that the distribution was much better with ICM. And so because it is so important to try to deliver as much virus as you can in a single administration, so because you're not giving it multiple times, they thought that delivery by the ICM route was very important. And they have a database of, I can't remember, hundreds of monkeys that they have dosed ICM, and that has been taken forward into clinical trials of gene therapy in other neurological disorders like frontotemporal dementia and Krabbe disease. and largely that administration procedure has been safe. What's important there is that you use a well-trained clinician, and the intent for Mavericks is to have the University of Pennsylvania carefully train all of the investigators in that trial. Lumbar puncture has been great for ASOs, but because you have to give it over and over, you wouldn't want to do that ICM.
Thank you.
Una curiosità sulla S2 e sui centri con cui collaborate, sulla geografia. Mi sembra tutto negli Stati Uniti come contesto. È perché in Europa abbiamo policy differenti e negli Stati Uniti è più semplice operare in un certo modo? Diciamo, siete interessati a allargare il confine, o è per prassi farmacologiche, perché lo sviluppo negli Stati Uniti ha modalità diverse da quelle in Europa? Diciamo anche i primi sviluppi sono meno globali, sono più concentrati sugli Stati Uniti, quindi c'è qualche ragione, un caso, o c'è proprio un'intenzione, diciamo, una concentrazione di Fast International più sugli Stati Uniti che sul mondo?
No, we would like to focus globally. So, for example, when we were developing the program for MVX 220, we met with the US FDA, but we also met with some regulatory agencies outside of the US in other countries. So we are in the process now of evaluating trial centers. So our intent would be to run studies in countries outside of the United States, because we think that it's very important as part of the development that we include other countries. We don't know what those countries will be yet, but we have definitely had conversations, including with some agencies outside of the US.
Okay, I don't see any other.
Thank
You
you.
have been asked so many questions. Thank you, Jennifer. Thank you. Here I don't have the cues from Benedetta, but I think we are ready to go up for lunch. They told me actually until 12:30 to hold you here because we were late. So, good. We can go up for lunch, and please let's come back by the time that you find in the program, so we start on time. Thanks a lot. Thank you to all the speakers. Thank you.